Now the time have reached to this point that the people are unable to afford the petrol of the car, and they are just going to have the alternate of the petrol so to drive the car. Now continuing the classification, so the basis of the classification is the spore formation. This is the sixth type of classification. Some bacteria can survive unfavorable and extreme condition by making the spores and producing the spore. These spores are the highly resistant structures formed when the nutrients are depleted, so they can live in those harsh conditions amicably till the time they are waiting for the good time to come, and they can flourish to make the bacteria again. These are seen in the gram-positive spore-forming Bacillus species, and like Bacillus cereus, anthrax, tetanus, and botulism. And botulism causing bacteria do have the spores, and they can resist all the harsh weathers. Here one can see the spore forming bacteria. These are the endospores, and here these are the vegetative cells. The spores under the favorable condition can change to the vegetative cells, and they divide and just multiply and increase their family. But the spores are the dormant. They can just live, and they are just waiting for the good time to become the vegetative cells. Here again one can see the spores by the stain, and that is just differentiating the spore. Now the another classification is oxygen requirement. Bacteria which need oxygen for their growth are called aerobic bacteria, and they can only flourish when they have got lot of oxygen available to them. Example is the Haemophilus influenza. But bacteria which grow well in absence of the oxygen are called anaerobic bacteria, and the oxygen is just like the poison for them. So in the oxygenated condition they are just going to kill themselves. So these are the bacteria which live in anaerobic condition. These are called anaerobic bacteria, and the example is the Clostridium tetani. As you have heard of the tetanus bacteria, which do grow in the wound because the wound is not having any proper oxygen. So that is anaerobic condition in which these bacteria grow well. Aerobic bacteria will die in the absence of the oxygen, but anaerobic bacteria will die in the presence of the oxygen, as I have told you. So the oxygen is the poison for these anaerobic bacteria. Now, the another classification is the host-parasite relationship, commensal bacteria. These are the bacteria whose natural habitat is the host. They live on the host only. They do not cause disease in their natural environment, like they live in the nasal passage of the infants which harbor the Haemophilus influenza. So these just have the shelter there, remain there, but do not cause any problem because it is their natural habitat to live in the nasal. Then symbiotic bacteria. These friendly bacteria which maintain a mutually beneficial relationship with their host in our intestine, like E. coli, they produce vitamin K. They are just like that any person who is just having living in someone's house and just paying the things to live there. So that is called the kind of guest who is living, but he is also paying for his food and another which he is using the things of that house. Then pathogenic bacteria. These are the bacteria which can cause disease like Clostridium tetani and Streptococci. Wherever they live, they cause the disease. Then cultural characteristic. Most can be grown bacteria artificially if atmospheric conditions and the temperature are correct. Difference in the effect of the oxygen on the bacterial growth. Classify the bacteria according to their requirement of oxygen. These are aerobes, which require free oxygen to grow. As I have told you in the previous slide, the anaerobes, which are unable to grow in the free oxygen, and they are killed if the oxygen is available. Then microaerophiles, as the name is, which grow best in the reduced oxygen concentration. Microaerophiles. Facultative anaerobes, which can grow only if oxygen is present, or they are just bound to grow if the oxygen is present or not. So facultative anaerobe, which can live when the no oxygen is available. Then classification on the biochemical properties, that is ability of the bacteria to produce certain enzymes like catalase, oxidase. On the basis of that, we can just recognize the bacteria. They are able to produce a certain enzyme. We do check and test these enzymes, and we can just say that these are this kind of bacteria. So bacteria are called catalase positive, which produce catalase enzyme, and these are the bacteria which are the staphylococcus. Or catalase negative, which do not produce catalase, these are the streptococcus bacteria. Similarly, certain bacteria produce oxidase. So these bacteria are called oxidase positive. These are the Pseudomonas, and there are certain bacteria which do not produce oxidase. These are called oxidase negative, as in case of E. coli. And here is the test, oxidase test. We just have certain chemical in which we put the bacterial culture, and with culture that shows the bubble, and when the bubbles are shown by putting the hydrogen peroxide on the bacterial colony, so we can say this is staphylococcus positive, and these are the catalase positive. These are staphylococcus which are catalase positive, and these are the stepped which are catalase negative. Similarly, one can see the oxidase positive and oxidase negative. One can see this is oxidase positive, and it is again here oxidase positive and oxidase negative. And very famous bacteria which is oxidase positive is the Pseudomonas. Then classification on the antigenic structure or the serology of the bacteria. Here different antigens are detected by the use of the specific antibodies, because certain bacteria produce certain antigens which can be detected by using the specific known antibodies which do react with these antigens, and we can classify the bacteria accordingly. Antigenic differences divide the members of any single species into subspecies or serovers. Examples: E. coli have the different antigens O, H, and K antigen typing that can tell the different subspecies of the E. coli, and similarly different types of the Salmonella and Enterica. There are 2, 4, 6, 3 types, and others that can be detected by the antigenic structure. Here one can see the subtype of the E. coli. These are the APEC, STEC, XPEC, and ETEC. These are E. coli, but due to their different antigenic structure and behavior, these are classified. Here, the people, those who are having the few hairs, like me, they can use this kind of method to get their cutting. Then another classification, the 12th type of classification, is the genetic composition. That is also called the molecular method. Previously it was not available, but nowadays PCR and other research that have made it possible to just classify the bacteria genetically, and these are the nuclear, these are also called the molecular method. There is the nucleic acid analysis, difference in the nucleotide sequence, and these are 16S RNA sequences, 23S RNA sequences to differentiate Shigella dysentery versus E. coli dysentery, those which look like similar, but because of their behavior of the different, as one can see the genetic composition. So from this genetic composition, one can see along with the other characteristic of the Shigella, that is going to just decide whether it is Shigella or the other bacteria. Here again one can see the sub units of the different bacteria. Here it is the 5S, here it is 23S, and combinedly they make the 70S, which is the characteristic of the bacteria, and these are having the different sub parts, and that can be detected by the genetic composition analysis. Then classification according to the phage type, that is to determine susceptibility of the bacteria to specific bacteriophage. These are also the new techniques which are being used nowadays to see whether the bacteria are able to susceptible to phage or not. So that is also one way. It is used in the epidemiological surveillance of the disease caused by the Staph aureus mycobacteria, Pseudomonas aeruginosa, Vibrio cholerae, and Staph typhi. There are certain susceptibility to the bacteriocin is also used as an epidemiological strain marker. Nowadays, phage and bacteriocin typing have been replaced by the molecular method, which is more easy to do, and that have got the better result. But in the past, that was phage typing was being used, and here one can see the phage typing of the bacteria sensitive to the phage and those which are insensitive to the phage. So bacteria can be classified. As you know, phage is the virus, type of virus which is capable of infecting the bacteria. So that is the phage. Now, classification according to the flow cytometry. It is divided into two parameters, FSC, forward scatter parameters found along x-axis. This gives a relative size of the cells. It measures amount of the laser beam that passes around the cell and that have got the scatter. Then SSC, side scatter parameter found along y-axis. As you can see, the forward scatter is the forward scatter. It is along the x-axis, and this is side scatter, so it is on the y-axis. It measures amount of the laser beam coming from inside of the cell, and so the 15th classification is the classification on the disease they cause. It is divided into three main groups. That is the respiratory organism, which cause the respiratory diseases, the Bordetella, Haemophilus, that causing the whooping cough, meningitis, pneumonia. Similarly, the zoonotic from the animals, that is Brucella, then Francisella, and Pasteurella, and Yersinia, plague, cellulitis, which is from the, that was the light scatter in the previous, in which I have told you that the forward scatter and the side scatter, that the missing of the slide, but now you can see the forward scatter and side scatter. As you can see, the forward scatter which is going straight away from the cell, and the side scatter which comes out from the side of the cells. So that is also the parameter of just judging the which bacteria have this much scatter and which bacteria have this much scatter. The past is where you learn the lesson. The future is where you apply that lesson which have you learned in the past. Now, the another classification that I have told you that the classification on the basis of disease they cause, it is just the continuation of the previous slide in which the zoonotic and the respiratory. The third is enteric and genitourinary organisms. Escherichia coli urinary tract infection causes diarrhea. Enterobacter that causes urinary tract infection. Serratia that causes pneumonia. Klebsiella that causes pneumonia, urinary tract infection. Salmonella, as you know, notorious for typhoid fever and enterocolitis. Shigella that cause enteritis and enterocolitis, and Proteus is just able to cause a urinary tract infection, very notorious for that. Another classification of bacteria is phenotypic method by which we can just divide the bacteria. Gram stain phenotypic method mean by certain physical method by which we are able to divide the bacteria, that is on the basis of gram stain, special stain, morphology, size, shape, colony, and growth, cultural characteristic, biochemical reaction. I have told you the catalase and other serological testing, antigenic. Then there is genotypic method. What is the genotype of the bacteria? That is through the gene probe, sequences, rRNA, and PCR, which can tell that it belongs to which genotype. So bacterial classification, phenotypic, the morphology, the biochemical, I have already told you, and the genotypic pattern of the fingerprint-based techniques, sequence-based techniques. And that is all for bacterial classification. I hope you will like this lecture, and if you like this lecture, please prescribe it and share it with the friends. And for any question or query, do not hesitate to talk to me on my phone number. And if you don't like it, again tell me that what the improvements I can have in that to make it more convenient and just swallowable to you. Thank you for patient waiting.